Hi, I am Anshita Gupta with another interesting and learning episode of B.Pharma on the important unit of introduction to dosage forms in Indian system of medicine, which are the two very important units of the section of pharmacognosy and phytochemistry 4 of B.Pharm 8th semester. Let's start our episode while taking a look at what we are going to learn today. Preparation and standardization of some important dosage forms including aswas and aristas, preparation and standardization of some important dosage forms like avlehas and churnas, analytical specification of aswa and aristas, analytical specifications of churna, churnam, fine powder, kwatha churnam, coarse powder for decoction, conclusion. Preparation and standardization of some important dosage form including aswas and aristas. Aswas and aristas are a special kind of medicinal preparation which are made by soaking the drug either in the powder form or in the kasaya form in a solution of sugar or jaggery for a specific period of time for fermentation to generate alcohol. The alcohol thus generated acts as a preservative and thus facilitating the extraction of active principles contained in it. It is preserved for many years. Example, Darkarista and Kanakasava. Aristas are made with decoction of herbs in boiling water, while aswas are prepared by directly using fresh herbal juices. These are unique liquid dosage form that contain self-generated alcohol. Aristas are classical Ayurvedic preparations typically used as digestive and cardiotonics. Arista, which also known as fermented decoction, and aswa, known as fermented infusion, are considered as a unique and valuable therapeutics in Ayurveda due to their medicinal value, sweet taste, and easy availability. People are prone to consume higher dosage of these drugs for longer periods. The manufacture and sale of aristas and aswas occupies an important place in the Ayurvedic pharmaceutical industry. Now, method of preparation of arista and aswas. The basic equipment required for the preparation of arista are earthen pot, sufficiently large and strong with a glazed exterior or glazed porcelain jar of suitable size, a lid of correct size to close the vessel, a cloth ribbon to seal the vessel, a paddle-like stirrer, a clean cloth of fine and strong texture for filtering, a vessel for boiling the drug. The major components are divided into 4 types according to their specific role in the process. This includes the main herb from which decoction is taken out as the case may be. They yield drugs which are pharmacologically and therapeutically much important in the given medicine, and the name of these medicines is derived from these herbs denoting their importance. The flavoring agents are herbs beside contributing to the flavor of the medicine have their own pharmacological action too. The fermentation initiator provides inoculums for the fermentation to start. The medium of sugar is required for fermentation. If the drug is dry and is to be used in the preparation of aswa, it is coarsely crushed and added to the water to which the prescribed quantities of honey, jaggery or sugar are added. If it is an arista, a decoction is obtained by boiling the drug in the specified volume of water. The water used should be clean, clear and potable. Now, different parameters of standardization of aswa and arista. The parameters stability of herbal formulation, that includes pharmacognostic parameters, physiochemical parameters, phytochemical parameters, microbiological assay and chromatographic analysis. WHO, the World Health Organization guidelines for quality standardized herbal or Ayurvedic formulations. First is quality control of crude drugs, material, plant preparations and finished products. Second comes stability assessment and shelf life. Third, safety assessment, documentation of safety based on experience or toxicological studies. Fourth, assessment of efficacy by ethnomedicinal informations and biological activity evaluations. The bioactive extract should be standardized on the basis of active principles or major compounds along with the chromatographic fingerprints including TLC, HPTLC, HPLC and GC. It generally involves the following parameters. First, organoleptic parameters. It includes color of sample. It generally includes order of sample, taste of sample, determination of pH of sample. Physical parameter includes determination of foreign organic matter. These parameters are for standardization of drug material. Second comes determination of ash value. It includes total ash value, acid insoluble ash, water soluble ash, sulfated ash. Next to this comes determination of extractive value. It includes alcohol soluble extractive value, water soluble extractive value. Next to this comes determination of moisture content and then determination of physical constant. It includes melting point, boiling point, refractive index, optical rotation. Determination of specific gravity, determination of solid content and determination of alcohol content. Now comes chemical parameters. Following parameters involves in chemical evaluation like alkaloids, drag and drop test, glycoside, Molish test, flavonoids, Shinoda's test, phenolic, lead acetate test, tannins, ferric chloride test, steroids, sulfoxide reaction, amino acid, ninhydrin test, carbohydrates, Fehling test and Benedict test. Fourth to this comes toxicological parameters. It involves following parameters. First is pesticide residue, second is heavy metal and third is microbial contamination. Arishta and Aswa are considered as one among best formulation in Ayurveda as they possess self-generated alcohol which act as self-preservative. The main difference between Aswa and Arishta lies in the preparation of kasya with few exceptions. Drugs used for preparing Arishta are found to be kanitha or madhyama, whereas in case of Aswa are muridu or volatile in nature. Aswa and Arishta are very popular forms of Ayurvedic system of medicine because of certain special properties which make them more beneficial than other preparations. These are nothing but the fermented syrups. These are the oral Ayurvedic formulation has been used as tonic since many decades. These formulations have been proved its importance and vitality as a tonic in many physiological conditions. So, its standardization has its own importance to check many of its properties as an oral formulation for its stability perspective. Now we are coming to preparation and standardization of some important dosage form avlehas and churnas. Avleha kalpana, it is another type of semi-solid preparation which is prepared either by adding jaggery or a sugar and is boiled with a prescribed drug, decoction of drug or its juice to get a semi-solid preparation known as avleha. Example, Kusmanda avleha. Avleha or leha is a semi-solid preparation of drugs prepared with addition of jaggery, sugar or sugar candy and boiled with prescribed juices or decoction. These preparations generally have kaya or other liquids, jaggery, sugar or sugar candy, powders or pulp of certain drugs. ghee or oil, and honey. Jaggery, sugar or sugar candy is dissolved in the liquid and strained to remove the foreign particles. This solution is boiled over a moderate fire. When pressed between two fingers, if paka becomes thready or called as tantuvat, or when it sinks in water without getting easily dissolved, it should be removed from the fire. Fine powders of drugs are then added in small quantities and stirred continuously to form a homogeneous mixture. Ghee or oil, if mentioned, is added while the preparation is still hot and mixed well. Honey, if mentioned, is added when the preparation becomes cool and mixed well. The leha should neither be hard nor a thick fluid. When pulp of the drugs is added and ghee or oil is present in the preparation, this can be rolled between the fingers. When metals are mentioned, the bhasmas of the metals are used. In case of drugs like bhalataka, a purification process is to be followed. The leha should be kept in glass or porcelain jars. It can also be kept in a metal container which does not react with it. Normally, lehas should be used within 1 year. Next comes churna kalpana. Churna is a powder of a single drug or a mixture of 2 or more drugs. Here, the drugs are finely powdered and they are filtered through a cloth so that the powder is very fine, which can be used internally as well as externally. This can be used for preparations like vati, avleha, etc. Churna is defined as a fine powder of drug or drugs in Ayurvedic system of medicine. Drugs mentioned in Patha are cleaned properly, dried thoroughly, pulverized and then sieved. The churna is free flowing and retain its potency for 1 year if preserved in an airtight containers. Triphala churn, Trikatu churn, Darkshadi churn and Sudarshana churn are some of the examples. Churna formulation are similar to powder formulation in allopathic system of medicine. In recent days, churna is formulated into tablet in order to fix the dose easily. These forms of medicament are prescribed generally because of their particle size. Smaller the particle size, greater is the absorption rate from GIT and hence the greater is bioavailability. A blend of several herbs and species make up the powdered mixture known as churna. Depending on its intention in medicinal, beauty or culinary use, its recipe varies. Common churna ingredients include cinnamon, fennel, dried ground ginger, coriander and turmeric. In the Ayurvedic diet, churna supplements are relied upon for many dietary and nutritional uses. The herbal mixture is thought to purify the blood and improve digestion when regularly used. Many Ayurvedic practitioners also use the blend to help prevent or treat inflammation. The immune system may be improved by ingesting these herbs as well. For example, Triphala churna is a blend of 3 Indian fruits. This include amla, baheda and harda. It has been used to help people control their diabetes. Excessive gas, eye disease and constipation have also been treated with these dietary supplements. Powder form is typically used in this case. Some people with asthma have found relief within the remedy. Several Ayurvedic recipes call for the remedy. Various herbal and fruit extracts are sometimes combined with the herbal mixture and sugar to create cough syrup elixirs known as Sitopaladi churna. Such solutions have also been known to be helpful in treating conditions such as indigestion, body weakness and chest congestion. Some people use the medicine for allergies as well. Ensuring safety of the indigenous system of medicines, there are adequate provisions under the Drugs and Cosmetic Act 1940 and Drugs and Cosmetic Rules 1945 for the regulatory framework and for monitoring the quality, safety and efficacy of drugs belonging to Ayurveda, Siddha and Unani systems. Licensing authorities are appointed by the state governments to oversee the enforcement of legal provisions for the manufacture and sale of ASU drugs. Good manufacturing practices and adherence to standards of drug as prescribed in the pharmacopoeia are mandatory for the manufacturing of licensed products to ensure quality, safety and efficacy of ASU medicines. Ayurveda, Siddha, Unani Drugs Technical Advisory Board and Ayurveda Siddha Unani Drug Consultative Committee are statutory bodies under the Drugs and Cosmetic Act to advise the central and state governments on technical matters and for securing uniformity throughout the country in the administration of the Act. Department of Ayush has implemented a centrally sponsored scheme of quality control of ASU and H drugs for strengthening of state pharmacies, drug testing laboratories and enforcement mechanism. Modern techniques helpful in identification and characterization of product of Indian medicinal system. Here first comes HPLC. The preparative and analytical HPLC are widely applicable in pharmaceutical industry for isolating and purification of herbal compounds. There are basically two types in preparative HPLC. Those are low pressure HPLC, typically under 5 bars, and high pressure HPLC, pressure bars greater than 20 bars. The most important parameters to be considered are of resolution, sensitivity and fast analysis time in analytical HPLC. However, both the degree of solute purity and the amount of compound that can be produced per unit time is recovery in preparative HPLC. The main aim is to isolate the herbal compounds, where as in analytical work the aim is to get the information about the sample. The preparative HPLC is the closest to analytical HPLC than the traditional PLC because it is having higher column efficiency and faster solvent velocities permit more difficult separation to be conducted more quickly. This is most important in the pharmaceutical industries because newer formulations have to be introduced in the market as early as possible. Now we come to high performance thin layer chromatography, also known as HPTLC. TLC is the common fingerprint technique for herbal analysis. The herbal compounds can easily be identified by TLC. In this technique, the authentication of various species, evaluation of stability and consistency of their preparations from different manufacturers. HPTLC is the common fingerprint mainly used to analyze the compounds which is having low or moderate polarities. HPTLC technique is widely used in the pharmaceutical industry for process development, identification and detection of adulterant, substituent in the herbal products and also help in the identification of pesticide content, mycotoxin and in quality control of herb and health products. Third one is gas chromatography, mass spectroscopy, generally known as GCMS. Gas chromatographic equipment can easily interface with a rapid scan mass spectrometer of various types. The flow rate of the capillary column is generally low, but enough that the column output can easily fed directly into ionization chamber of mass spectrophotometer. In this, the simplest mass detector in GC is the ion trap detector. The ion trap detector is remarkably compact and less expensive than quadrupole instruments. It has been used for identification of large number of components present in natural and biological systems. Coming to next point, we are having liquid chromatography mass spectroscopy, commonly known as LC-MS. LC-MS is the one of the most prominent method of choice in many stages of drug development. The chemical standardization of an aqueous extract of the mixture of the herbs provided chemical compounds serving as reference marker using LC-MS. A good example or application of LC-MS can be observed in the pharmacokinetic studies of Chinese medicinal herbs. Interference peaks in biological samples are easily observed when using HPLC coupled to ultraviolet, fluorescence and electrochemical detectors. Coming to next point, supercritical fluid chromatography. The supercritical fluid and microbore liquid chromatography offer potential applications for the drug analysis. In this, the mobile phase is a gas, commonly used CO2, maintained at its supercritical state, that is, above its critical temperature and pressure. The SFC mobile phase has low viscosity approximating that of gas and a high diffusivity between those of capillary gas chromatography and liquid chromatography. There are various other studies also which help in identification and authentication of a particular medicinal system about its purity and safety. An interesting example of bhasma can be discussed here. Bhasmas are the Ayurvedic metallic preparation in which metal act as a nanocarrier for drug delivery and are widely recommended for treatment of a variety of chronic ailments and are taken along with milk, butter, honey or ghee to eliminate the harmful effects of metals and enhancing their biocompatibility in the body. Neutron activation analysis of 20 metallic based bhasmas such as calcium, iron, zinc, mercury, silver, potassium, arsenic, copper, tin, gemstones confirmed the purity of these bhasmas as the other elements such as sodium, potassium, magnesium, vanadium, manganese, iron, copper and zinc were found in microgram per amount and aluminum and cobalt in ultra trace nanogram per gram amounts. Various techniques like atomic force microscope, scanning electron microscope, transmission electron microscope and energy dispersive spectroscopy have been employed for the estimation and characterization of bhasma. The Kaisa bhasma analysis qualitatively through X-ray diffraction. Fourier trans-infrared spectroscopy correspond to inorganic metal, hydrated metal salt or oxide. The FTI spectra showed no peak for any organic molecule or bond corresponding it, thereby confirming the absence of organic matter and external organic contamination. The AAS, also known as atomic absorption spectroscopy, study was conducted to determine the concentration of elements present in the formulation. The results showed that the element iron was seen in the major concentration of 85.91%. AFM analysis confirmed the formulation has spherical morphology with an average particle size of 100 nm. The spherical morphology was due to the aggregation of the nanocrystals of the metallic oxides. It has been used in traditional medicines for treatment of anemia, hepatotoxicity. If the herbal or traditional system of medicinal products are evaluated and analyzed using sophisticated modern techniques of standardization such as UV visible, TLC, HPLC, HPTLC, GC mass, spectrophotometric and other methods, the work of regulatory authorities, scientific bodies and industry in the development, assessment and registration of such product will become easier and widely acceptable. Now comes evaluation parameters as per pharmacopoeial standard of Ayurvedic drugs. Standard of identity, purity and strength. Comes foreign matter, total ash, acid insoluble ash. Next to it comes alcohol soluble extraction. To this include TLC, known as identity test, with method and description, assay method of major chemical constituent, properties and action, rasa means taste, guna properties, virya predominant action, vipaka metabolic action, karma pharmacological action. Next to it comes toxicological assessment. First to it is test for heavy metals, second is microbial load, third is important formulations, fourth is therapeutic uses, fifth doses, sixth is authentic textual references. Analytical specifications of Aswa and Aristha fermented liquids. First is specific gravity at 250 degrees Celsius. Second, alcohol content. Test for methanol should be done. Third is total acidity. Fourth is non-reducing and reducing sugar. Now comes module 5.1, analytical specifications of churna, churnam, known as fine powder, kvatha, churna, coarse powder for decoction. It includes loss on drying at 105 degrees Celsius, particle size between 80 to 100 mesh for churna, 40 to 60 mesh for kvatha churna. Module 5.2, acceptable limits for products, parameter and its specifications. First comes microbial contamination. Total bacterial count 1:10⁵ CFU per gram. Yeast and mold 1:10⁵ CFU per gram. E. coli should be absent. Salmonella absent. P. aeruginosa absent. S. aureus absent. Second comes pesticide residue less than 1 ppm. Heavy metals, lead should be 10 ppm, mercury 1 ppm, arsenic 3 ppm, cadmium 0.3 ppm. Aflatoxin B1 should be 0.5 ppm, G1 0.5 ppm, B2 0.1 ppm and G2 0.1 ppm. Students, the figure shown below shows different standardization techniques that could be counted when the quality of a herbal or Ayurvedic dosage form is concerned. The figure shown here clearly represents the different techniques that is accounted for standardization. First is organoleptic nature, botanical description, chemical nature, biological nature and physical nature. Coming to the last module, conclusion. It is a well-known fact that traditional system of medicines always played important role in meeting the global healthcare needs. They are continuing to do so at present and shall play major role in future also. The system of medicines which are considered to be Indian in origin or the system of medicine which have come to India from outside and got assimilated into Indian culture are known as Indian system of medicine. India has the unique distinction of having 6 recognized system of medicine in this category. They are Ayurveda, Siddha, Unani and Yoga, Naturopathy and Homeopathy. Medicinal plants based traditional system of medicines are playing important role in providing health care to large section of population, especially in developing countries. Interest in them and utilization of herbal products produced based on them is increasing in developed countries also. To obtain optimum benefit and to understand the way these systems function, it is necessary to have minimum basic level information on their different aspects. Indian system of medicine are among the well-known global traditional system of medicine. In this review, an attempt has been made to provide general information pertaining to different aspects of this system. This is being done to enable the readers to appreciate the importance of the conceptual basis of this system in evolving the material medica. The aspects covered include information about historical background, conceptual basis, different disciplines studied in the systems, research and development aspects, drug manufacturing aspects, and impact of globalization on Ayurveda. In addition, basic information on Siddha and Unani system has also been provided. Here, we come to an end of this lecture session. Keep conceptualizing the matter. I'll be back with next lecture on general method of extraction, isolation, identification and characterization of phytoconstituents of carbohydrates, alkaloids and glycoside. Till then, keep studying. Goodbye.